Artificial intelligence is changing science at an unprecedented rate. It's not just AI tools for scientists to use in the lab, but increasingly ways of discovering new and better drugs.
90% of drugs fail, and the few that survive are incredibly expensive, cost around $6 billion per novel approved drug.
There's all kinds of reasons why someone responds well to a drug and why someone doesn't. But one thing that people don't know how to model at all is how the gut is involved in that.
This is David and Arvid, the two co-founders behind Bio, and they're building an AI engine that is not trained on understanding images and texts or writing your next email, but it's trained on the data of what is happening in the tiny little creatures that live inside our guts, our microbiome.
You'd expect that this only plays a role for drugs that you take orally, right? Because the microbiome, it lives in the intestine. So why would ever like a drug you would, for example, inject end up interacting with a gut bacteria?
David went on to give me this example. Some of the most common chemotherapeutic agents are injected into the bloodstream. However, they get metabolized at the liver, at which point the metabolites or waste products are excreted into the gut through the bile duct. Different patients will have different microbiomes that process these waste products a different way. And for some patients, this chemotherapeutic drug might get reactivated and back into the bloodstream.
So basically, the problem with the microbiome is that it's incredibly complicated and contains a lot of data. The new drug you came up with, to giving it to someone and then basically exposed to their very complicated digestion and how the microbiome interacts with the drug. There's basically so much black box in that step that messes with the success rate of drugs. And we looked at a lot of different things in the drug development process so far.
But here's the problem. Many different things make up your microbiome, each one at different concentrations, different levels, and with different gene expressions. And although we have been collecting all this data for a really long time, it is really difficult to make sense of it.
What I really care about is learning from all the past things we've done, essentially around drug development. We came up with a lot of drugs, we gave it to a lot of people, we sequenced tons of people, we have enormous repos of existing data, but we can't do anything with that. If we only could get the insight from all this stuff, we'd essentially do 10x better. It's basically like not having history books, never reading books and essentially repeating all the same mistakes all over again.
With mountains of data from previous clinical trials and a deep understanding of the microbiome profile of the patients that received them, Bio is going to be helping the right patients get the right drugs.